everyone. I hope all of you are doing pretty fine. And today we are going to finish this chapter. So yay. Okay. So in this chapter, we have discussed so many things ranging from how exactly there are different type of cells, how exactly the functions and shapes of these cells basically are correlated or interrelated, the different organelles of cells, the different type of organisms on the basis of number of cells, the differences between the prokaryotes, eukaryotes, animal cells, plant cells, right? So we're just going to finish up a bit. Okay, we're just going to touch up those topics a bit and then this chapter will be finished. So moving forward, we basically are be going to have the topics structure of animal and plant cell in this particular class, prokaryotic, eukaryotic cell, and cilia and flagella. Now listen to me very carefully. Most of the questions asked are asked from this particular topic. Okay, although we have discussed both the structure of plants and animal cells well in advance and the prokaryotic and eukaryotic cells as well, but we're just going to touch up those differences a bit and in just a little more detail. Okay, although I did give you the difference table between the plant and animal cell in the previous session as well, so you might as well just go to that. Okay, that's like the easiest and most contentful table. And then we'll be doing the cilia and flagella, which could be the star of today's session because this will be the completely new topic for all of us. Okay, like for you, obviously, not me because I'm the teacher, duh. Anyway, moving forward, we have the structure of animal and plant cell, right? See, structure of animal and plant cell, we did discuss how there were the cell wall and chloroplast and vacuoles, the three organelles which were present in the plant cells and which were completely absent in the animal cells, except the vacuoles, which could or could be, which could or could not be present. And if they were present, they were in very small size. Okay. Apart from that, we did talk about these centrioles as well, which were present in the animal cells and absent in the plant cells. Remember centrioles, which basically were used for the sake of division of chromosomes during the cell division. All right. Now, that being said, we also have the structure here, the diagrams here for both of like plant and animal cells. Okay. We'll just move forward to one, like both of them one by one, and we'll be revising whatever we did in the previous session. And then we'll be moving forward to the prokaryotic and eukaryotic cell. Okay. Right. So first, let's just go by the keywords in this particular explanation. Plant and animal cells are very similar. Okay. Why? Because both of them are eukaryotic cells. Eukaryotic, true nucleus. Okay. Both contain membrane-bound organelles. Both have the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and peroxisomes. But the cell wall and chloroplast and large vacuoles, large central vacuoles are present in the plant cells. Okay, whereas in the animal cells, we have smaller vacuoles and centrioles, right? I'll write this down here for you once again. Used in cell division. Used in cell division. Right? Now that is one thing. We'll just go over by the entire structure of plant and animal cell once. So the plant cells and animal cells, we will be discussing the organelles which are different in both these cells, because otherwise the similar organelles we have already discussed in the previous sessions in detail, along with the structure and other characteristics of those organelles such as the mitochondria and the Golgi apparatus, endoplasmic reticulums and lysosomes, etc. Right? So you see, we have the cell wall in the plant cell, which is one structure, okay, which is absent in the animal cell, okay. If it is shown in the animal cell structure, this is incorrect, okay. Make sure that you learn that. Do not be fooled by that ever, okay, because the cell wall is present in the plant cell, algae and bacteria. It is never present in the animal cell. Never ever, okay. Keep that in mind. Apart from that, we have the chloroplast. Chloroplast is also the organelle which is present in the plant cell and not the animal cell. Why? Because it enables the plant cells to carry out the photosynthesis, which can only be done by the plant cell by capturing the sunlight, right? Apart from that, we have the vacuole. Vacuole is also shown in the plant cell structure. Why? Because plants require the large number of vacuoles, or plants require the large sized vacuole in order to store the water and nutrients and food and the secondary products, etc., etc. And that is why their vacuole is present in the center and their nucleus is present in the sideways, okay, not in the center. And on top of that, we have the centrioles, which are just present in the animal cells and not the plant cells. I would suggest you to go to the previous session and learn the plant and animal cell structure and differences from there in detail. Okay. For now, we're just going to move ahead to the prokaryotic and the eukaryotic cell. Now, listen to me very carefully. We did talk about the prokaryotic and the eukaryotic cells, how the prokaryotic cells are the ones which basically are the primitive cells or the old cells or the ancient cells. Okay, why? Because they had been present on the surface of earth for like millions and millions of years. On top of that, they have never evolved, they have never evolved after that. Okay, once they were made, they were just made, they have not evolved after that. Whereas the eukaryotic cells are the advanced or the modern cells which have evolved according to the environment and are so much better at surviving, right? Now, see, prokaryotic organisms are the ones which have single prokaryotic cell. When I say single prokaryotic cell, that means the bacteria are single celled organisms. Okay, they are not multicellular organisms, they are unicellular organisms. The prokaryotes are unicellular organisms. I'm just going to write that down here for you. Okay. Apart from that, the eukaryotic cells are found in the plants, animal, fungi, protists. Okay. Listen, in the ninth standard, you'll be learning about the five kingdoms in the entire living world. Okay. The protista, the Monera, Protista, Fungi, Plantae, Animalia. Okay, Monera is called as the prokaryotic kingdom because it has all the prokaryotic organisms arranged in it. Then we have the Protista. Protista is the only kingdom which has unicellular eukaryotic organisms. And then fungi, plantae, animalia are the three different kingdoms which have multicellular to unicellular eukaryotic organisms. Okay, and they're differentiated on the basis of several features such as their mode of nutrition, their mode of reproduction, etc., etc. All of this will be learning in detail in the ninth standard in the chapter diversity in the living organisms. Okay, that's a long chapter, but it is super interesting. Now back to the prokaryotic and eukaryotic cell. The prokaryotic cell are smaller and eukaryotic cell are larger. Why? Because the prokaryotic cells have to accommodate a large number of organelles, whereas the eukaryotic cells, they do not have to accommodate any kind of organelle in the first place, or any kind of advanced organelle in the first place. Moving to another difference, the DNA is contained within a membrane bound nucleus in the eukaryotic organisms. Eukaryotic organisms have the DNA enclosed in a nucleus. True. Karyotic nucleus. I think I've told this to you multiple times, like countless number of times. Okay. Pro primitive or old karyotic nucleus. Okay. Now, that being said, what you see right here is we basically have the bacterial cell. Okay. We have bacterial cell right here. And this is any kind of animal cell which could be present as a eukaryotic cell. It is very important for you to understand that there are several differences based upon the cell size, nuclear membrane, etc., etc., in the prokaryotes and the eukaryotes. Okay, so I'm going to give you a table and I'm going to explain that to you in order to make the understanding a little better and clearer. And in case of any problem, you can just ask your teacher, which is assigned to you for biology. Okay. And okay, now moving forward to this one, important thing what you see right here in the prokaryotes and the eukaryotes, as I told you, prokaryotes are mostly smaller and eukaryotes are mostly larger. Why? Because they have to accommodate different cell organelles. Okay, what are cell organelles? Cell organelles are the part of cells which help in the survival. For example, the endoplasmic reticulum, Golgi apparatus, lysosome, ribosome, etc., etc. Right? Apart from that, capsule. Now listen to this. Capsule is the most interesting feature which is possessed by the prokaryotic cells. Okay, and it is completely absent in the eukaryotic cells. Remember when I told you that prokaryotes are the bacterias? The bacterias can or cannot have a capsule. What is a capsule? Capsule is like the outer covering of the entire cell, which is very, very shiny, which is very, very slippery. Okay, and it is made up of the chemical which is called as the mucopolysaccharides, kind of the carbohydrates. Okay, now the bacteria which will have the capsule are pathogenic. Now, when I say pathogenic, that means they are more than capable of disease being caused to the person wherever or whoever they are present in. Okay, next is the cell wall. As I told you while teaching the cell wall, that the cell wall can be composed of different kind of materials according to the organisms. I did give you the example of the plant cell, which was the cellulose, hemicellulose, pectin, lignin, etc., and the fungi, which was made up of the chitin. Right now, the cell wall which is present in the prokaryotes, they are also made up of carbohydrates, whereas the eukaryotes could be plants and fungi. They have made up of cellulose and hemicellulose and chitin in the fungi. Okay, now nuclear membrane is absent in the prokaryotes but is present in the eukaryotes. Vacuoles absent, ER absent, mitochondria absent, Golgi apparatus absent, and lysosomes absent. All this suggest the smaller size, and hence the smaller size, because they do not have to accommodate anything. Okay. Next, we have the ribosomes. Remember when I told you that ribosomes basically are the protein making machineries, and ribosomes are of two types, the 70S and 80S. S is the sedimentation coefficient. It is also called as the Swedberg's unit. It basically is determined on the basis of settling it under the under the influence of centrifugal force and gravity, right? Ribosomes, the 70s are the smaller ones which are present in the prokaryotes, and 80s are the larger ones which are present in the eukaryotes, and they help in making up of proteins with the help of mRNA, which is called as the process of translation. I did tell you about that if you have listened to all the sessions very, very carefully. Okay, and if not, go back, revise, and then come back here. Okay, moving back to the chromosomes. Remember when I told you that chromosomes are the condensed form of DNA when we were doing the nucleus and when we were doing the centrioles? I did tell you that chromosomes are the super folded or the condensed form of the genetic material, which is the DNA. Okay. Now see, chromosomes, basically true chromosomes are present in the eukaryotes. Why? True karyote nucleus, they have a true nucleus, so they'll have the true chromosomes as well. Okay, in the super folded form and super arranged form, whereas the chromosomes in the bacteria, they could be absent. Okay, and the DNA could all be, lying in an unfolded state, in let's say lazy or messy state. Why? Because they do not have the protein. Now if you remember correctly, I did tell you about the histone proteins. Those histone proteins help in the super folding of DNA into the chromosome. So I'm going to write that down here for you in order to avoid any confusion. So they help the DNA to superfold in the chromosomes, and that is how the chromosomes look. They have a butterfly structure. Okay, and they're held together by the proteins adhesively. Now see, they do not have the histone proteins, so the chromosomes could or could not be present. That completely depends upon the organism that we're talking about. And then we have the division. Division basically pertains here to the cell division. Okay, now when I say cell division, there are multiple type of cell divisions. There is mitosis, there is meiosis. Mitosis basically leads to the formation of the cells which are identical. Meiosis leads to the formation of the germ cells, which are the sperm and the eggs, right? Apart from that, important thing right here is that the simple fission is what we are talking about, the division in identical cells, identical daughter cells, right? And here the division is because of the spindle of microtubules. The spindle of microtubules are the ones which are made up by centrioles or produced by centrioles in order to divide the chromosomes into the two halves so that they could be separated and distributed into the two daughter nuclei separately and equally. Let me just draw that out here for you. Suppose this is the parent cell and these are the chromosomes which are arranged. Okay, so what will happen, these are the centrioles which have moved towards the opposite poles, and they'll start producing the microtubules or the spindle fibers like this. Okay, and those spindle fibers, these are called as the spindle fibers which are made up of microtubules. Okay, so what will happen, microtubules could be anything. They could be made up of actin, or they could be made up of like actin basically. Okay, right, back to this. Actin is a protein. Back to this. These are the chromosomes. Now what will happen, these, look at my video, these will just start contracting, and these will start separating the chromosomes in the opposite direction in order for them to be distributed into the two daughter nuclei. Okay, next we have is the meiosis. Meiosis is basically present in the multicellular organisms. Now when I say multicellular organisms, it leads to the formation of germ cells. What are germ cells? Germ cells are the ones which take part in the reproduction process, for example, the sperm and the egg. Right, now that being said, it is absent in the prokaryotes, it is present in the eukaryotes. Okay, because the eukaryotes are the only cells which make up the multicellular organism. That is all about the prokaryotes and the eukaryotes. Next thing that we have is the cilia and flagella. Now, see, cilia and flagella are the kind of cell organelles which help in making a cell motile. Now, when I say motile, that basically means they help in the cells moving from one place to the another. Now, cilia and flagella, they are present in the multiple cellular organisms. They could be present in both prokaryotes and the eukaryotes. Okay, these are the projections from the cells. Okay, projections mean that these are the outer growths. Outer growths. Okay, now see, they are motile as in they help in the making the cell movable, and they're designed either to move the cell itself or move the substances over and around the cell. Okay, they also help in the capturing of the food, and they'll be moving the food in this manner towards the cell so that cell can be fed. Next thing is the primary purpose of the cilia and the mammalian cells. When we are talking about the mammalian cell, we could be talking about the human beings. Okay, let's just talk about the human beings first. Human beings, right, they help in moving the fluid mucus cells over their surface, etc., etc. See, remember this, you have gone through the chapter how do organisms reproduce, or if you've not, you'll be just going through it in the standard itself. So there you'll be learning about the structure of the female reproductive system. And the female reproductive system, you know that there is an ovary and there's the fallopian tube or the oviduct like this. Okay, now inside this oviduct, this is the oviduct, this is the ovary which produces the egg, this is the uterus, okay, and this is the cervix, and this is the vagina. Right, now what will happen, this is the female reproductive system, FRS. Now what will happen, since the egg is produced from here, the egg will have to travel to the oviduct. How will egg travel to the oviduct? If we look up into the internal structure of the oviduct will have the cells like this. Okay, it will have the cells like this, and over the cells there'll be hair like projections or the cilia which will be present like this. Okay, and these cilia, they'll help in moving the egg from this direction to this direction towards the uterus. Okay, towards the uterus. So that is how they help in the movement of different substances in the mammalian cell. Okay, see, they both the cilia and flagella, they have the same internal structure. Okay, and they move in certain directions. I'll give you how exactly they move. Okay, and they could be present in the bacteria, they could be present in the eukaryotic cells as well. I think I did tell you about this previously. Okay, now moving forward, this is how they move. This is the movement of the flagella. This is the movement of the cilia. Now see, difference is that flagella is a larger structure and cilia are the small hair-like structure. Flagella could be present in the fewer numbers and cilia always have to be present in the larger number in order to have their effect in let's say in order to have their effect, okay, since they are smaller in size. So I'm going to write that down here for you. Flagella are larger or whip tail like structures, whip tail like structures, whip tail like structures, okay, and they are present in fewer numbers, okay, fewer numbers. Whereas if we talk about the cilia on the other hand, my friends, are small hair-like structures. We'll be talking about their movements as well. Don't worry about it, okay? So small hair-like structures, and these structures, they are present in larger amounts, and these cilia, they move back and forth and the flagellum move in a proper propeller-like motion. Now look at my video. I'll tell you how the flagellum moves. Suppose this my hand is the flagella, like my hand is the bacterial cell and my finger is the flagella. Okay, the finger is attached to the bacterial cell. The flagella is attached to the bacterial cell. So in order to cell to be moving forward, the flagella will move like this, okay, in a circular motion, just like a propeller. Okay, I hope you can see this. Okay, but if we talk about the cilia are going to move like this. Okay, I hope this is clear enough since you guys have me dancing. Anyway, now back to the topic. I mean, this is all about the chapter that you need to know. And now I've got a question for you. Okay, question is, which cell has a larger vacuole, or out of pro and eukaryote, which one will have which one will have which one will have the endoplasmic reticulum? Okay, now I know that you know the answer, so I'm giving you like 5 seconds to answer the both questions and then we're going to discuss these. Okay, time's up. See, which cell has the larger vacuole? Plant cell. Out of which prokaryotic and eukaryotic cells will have the endoplasmic reticulum? Remember, the endoplasmic reticulum is a double membrane bound organ. Okay, no, the endoplasmic reticulum is present in the eukaryotic cells because it is present in continuation with the nucleus and it is responsible for the production of the lipids and the proteins, and that is why it is present in the eukaryotic cells, eukaryotes. That being said, this is the end of the chapter and thank you so much. And you guys take like a lot of precautions, stay safe and happy and learn a lot. I'll see you in the next chapter maybe. Bye bye.